know how to get the screen to show too.
Okay. I just wanted to make sure I could get this going, but it's going. It's just super laggy.
Okay.
Because look, this is you walking over. Isn't that so creepy?
That's really weird.
But this one is live.
Oh, okay.
So, like, is that okay?
I think that's good, yeah.
Cool. Jordan, do you want to check on your phone if it works? Thank you. Oh, Phil just texted. Your talk is less than an hour, right?
Yeah. It's 15 minutes.
15?
50.
It should be.
Oh, I can record it. Would you like it recorded as well? Oh, I can do that. It'll be on YouTube after, online, but I'm also now recording it, I think, to my laptop.
Okay.
I could be wrong about that part.
Cassie didn't think it was going to be on.
No, I didn't know.
It's okay, we got your back. Just to see yourself, or do you want me to make it so you don't see yourself?
Maybe not. Beautiful.
Should I sit here to make sure it works the whole time?
Sure. It's already so
How's your day been?
full of anxiety. Trying to remain calm and be busy.
I like your suit choice. You look very professional. It was far too cold to wear a dress. Remind me, I have to show you a progression of photos later.
Of what?
Of my experience last night / this morning, mentally preparing for this defense.
Oh my god.
There's 5 people already on the screen.
See?
It's a lot of
Looking at you
pressure.
right now.
There's 7 people watching right now, Cassie.
We're
is that?
8.
on the computer.
We have 1 like.
One light. Wow.
Because you don't want, because the link is public, so we don't need like an account to
Oh,
watch
that's true.
Yeah. I thought about making it private, but I figured it was okay. I just copied what Alex had.
It's fine.
Phil confirmed that it works. And also that my two Wi-Fi.
Very good.
We shared it with like 10 people, so.
Excellent.
I mean, I don't imagine that randos go on and look at people's thesis defenses.
Well, I had a panic this morning because to live stream from a YouTube account, you need to get it approved, and it takes 24 hours. And I, of course, left this till this morning to figure out. So I'm like freaking out that I didn't have 24 hours, so it's on Greg's account. So Greg gave me his like login info and password. So it's from Greg Jones's account.
Save the day.
Truly. And in like 23 hours, I'll be able to live stream anything I want.
Are you the musical accompaniment of the defense?
Oh, I wish. I'm in charge of the live stream, and I feel a lot of pressure.
You're doing a great job.
I hadn't realized that particular instrument existed in this room until now.
Yeah, I don't really come to this room very often.
I mean, mostly I come here for Cambio seminars.
Is that this floor? I thought that was 370.
No, I think it's this one.
Oh. Then I do come here.
This is fine with one screen, right? That's all I could get. So I tried the middle one, it didn't work. And I tried this one, it did work, and I tried both of them. Only worked on this one.
Would you like the other one?
No.
Okay. I think this is better.
That's only one.
Yeah.
Well, wrestler just counted 8 Cassie.
Oh, she signed it off Val.
That's cute.
Oh, we're down to 6 views.
Oh, losing people. Oh, they lost
Okay.
It's present in the moment.
2 likes though.
A lot of pressure. I'll just screw up.
Or is there a way to aim just like slightly up?
In like a minute, you'll see what I did. But now I don't see all of Cassie's there.
It's okay.
There you go. You can see it moving now. Is that better, Jordan?
Hold on.
It still hasn't moved for you?
It's a 15-second
It
delay.
doesn't.
Yeah.
Like it's just a smidge back down.
I did already. Did you see me move it a second time? Is that okay?
So we'll be broadcasting the video of you saying testing soon.
That one got deleted already. I've also been requested to live stream the toast portion if that's alright with you.
That's perfectly fine.
Hopefully the audio is okay. Wait, Cassie, say something?
Hello. I like that was kind of quiet. I'll talk louder.
I could hear you right there.
Okay.
I can hear you.
I'll
Cool.
project when
The
I
audio
start.
mixer bars are moving as we speak, so it's picking up something.
Okay. I'll try to project more. I should have turned my phone off.
Yeah, just loads of time.
What?
Have you ever seen the movie Zootopia?
No.
I've heard it's really good. Spencer was raving about it earlier.
Oh, okay.
You guys? Should be a good song for it.
Oh, okay.
It's on Disney Plus.
Great. Watch it later this weekend.
Have you heard from Laura today?
I don't know if she went to that so much. But 1:30
It got moved.
to 2.
I thought it got moved days, but if you maybe I made that up.
I thought you said it was 1.
Wait, if it got moved to 2,
That would be pretty tight.
Maybe I'm
As
wrong.
it is, I'm very worried.
I could be wrong.
probably
I'm sorry, Cassie.
It's okay, I'm gonna turn myself back on and double check. I just feel like I've done something. Made a mistake.
did your room get moved to the last? I
Not
shouldn't
that
say.
I'm aware of.
I really shouldn't have said that. My apologies.
No, it's okay. I feel like we're still on okay time.
I forgot a notepad. You were in the wrong room, Cassie?
I just wanted to double check.
That's okay.
Yeah. First one new, so.
Still?
Yep.
I'm feeling guilty. So.
No, you're great. Right on time.
Actually, technically, I'm in the early, so.
I just don't write the questions.
Look at Catherine. Look like her.
That was awful.
Oh, sure.
Can you turn and face us?
Doesn't feel very organized.
You're succeeding in smiling for the camera, Cassie.
It's very organic.
We're still waiting for ours. Oh, Shayla. We're just gonna have to ask. we started at 3:05, so we cool.
You're talking MIT Standard Time.
Yep. Oh, it's so mellow. It's unbelievable.
I want to make sure you're in the camera line.
Who is that for?
Everyone.
Who's watching?
Phil.
Who else?
Alex's mom.
Oh my gosh. Oh my gosh. Oh. Maybe there's stuff on my computer.
See it.
You see it? Okay. Oh, we're right on time. See,
Yeah.
we have time. So you have it all set up.
I think we're good.
All right, so welcome everybody to Cassie Jarvis's Thesis Defense. Before I let Cassie take the floor, I'll just tell you a little bit about her, some of which I just verified. Cassie grew up in Illinois, and then she was an undergraduate at Illinois College. And she had a perfect 4.0 there. She was a biochem major, but she tutored physics. So she's starting out with a pretty broad background. But she was really interested in research, and especially in research at the interface of chemistry and biology. And so she went over the summer and did a research project at WashU. And this was a special program for talented undergraduates. And it turns out she worked for Jim Janetka, who was, fortunately for me, a graduate, a PhD from Dan Rich's lab. And Dan Rich was at Wisconsin at the time. So we had an inside track, I think, to recruit Cassie to Wisconsin. And she worked on making inhibitors of carbohydrate binding proteins. Also good for me. So when she came to Wisconsin, I was able to recruit her to my lab, and she decided to take on this pretty challenging project that she's going to tell you more about. But what really impresses me with Cassie is just her fierceness as a scientist. She will just go into any new area and do any experiment that she needs to. So her background was largely in organic synthesis, but then she just, she was like, oh, I need to work with mice in order to really test my ideas. So she was doing that, and then she was like, okay, chickens, I'll be working on them too. And you'll hear a little bit about that. And the other thing I will say about Cassie is, and maybe this has to do with her fierceness, but she's also highly creative and just gets stuff done. So you always want her on your team. And she can be on your team at, 1 a.m. when you're contacting people in your lab, such as Marshid and Cassie, about a grant whose deadline you didn't realize and you're way behind. And that's that's an amazing thing because because that's really super duper extra credit for grad students. But you also want her on your volleyball team because she will show her fierceness there, especially if there are some lousy shots being made. So anyway, I think you're gonna hear some really cool science, and I mean, assuming all goes well today, I'm really gonna miss Cassie. So go ahead, Cass.
Well, thank you very much. Thank you so much for that kind introduction. I'm really excited to be here today and talk with you about some of the research I've done in the Keesling group that has largely focused on using polymers to target specific immune cells and probe different facets of immunity. And so today my talk is going to be divided into two different portions. The first, I'm going to be discussing how we use polymers to look at how the physical property of an antigen can have dramatic impact on trafficking through dendritic cells. And then in the second part, I'm going to switch over to talk about how we've been using polymers to look at how antigen display on a polymer backbone can really influence immunity, particularly B-cell activation, for a focus on informing vaccine design. So I'll just jump right into the background for the first part of the talk. So this is going to focus largely on dendritic cells, and we've been really interested in dendritic cells because of their unique role serving as a bridge between innate and adaptive immunity, where they function much like an innate immune cell where they're constantly sampling the environment, they can secrete cytokines, but then they also take up antigen and can present it to cells of the adaptive immune system and actually shape these responses via the cytokines they secrete. So dendritic cells are professional antigen-presenting cells, and as immature cells, they are constantly circulating in the periphery of the body, where they're sampling the environment and they're taking up pathogen and degrading it into peptide portions. And throughout this process, the dendritic cells begin to mature, and through maturation, they migrate to the lymphoid tissue, where they encounter cells of the adaptive immune system. And it's here that they can take their processed peptide cargo, present it on surface MHC molecules, and recruit that helper signaling. And so because dendritic cells play such a critical role in the immune system, they have to have mechanisms to be able to recognize a wide variety of pathogens and facilitate that recognition and uptake. And so they express a huge number of receptors on their cell surface to facilitate this. And we are a carbohydrate lab, as Laura mentioned, and so we really are interested in the carbohydrate binding lectin receptors that these dendritic cells express, and in particular one major class of C-type lectins. And so these lectins can recognize conserved carbohydrates on the surfaces of pathogens, facilitate that binding, and then pathogen uptake. And in particular, we've been really interested in this receptor DC-SINE, which can bind to high mannose and fucose-substituted glycans, and has been linked to a number of really important pathogens. And so here is sort of a handful of pathogens that have been linked to being involved in DC-SINE-mediated uptake, and you can see there are a lot of these that are very relevant for human health. And so while these interactions with pathogen and DC-SINE often produce productive degradation and immune presentation and activation, many of these pathogens have actually developed mechanisms where they can utilize this receptor for their own benefit and actually evade the immune system. And so HIV is a classical example of this, where when it interacts with DC-SINE and is taken up via DC-SINE, rather than going through this endocytic pathway for degradation and antigen presentation, it's actually developed mechanisms by which it can subvert endosomal routing and instead go to these invaginated compartments that are near the cell surface. And it uses these compartments where it hides out and remains undetected, and then essentially uses the dendritic cell as a Trojan horse to hitch a ride to the lymphoid tissue where it can see its target CD4 positive T cells and then easily pop out of these pockets to propagate infection. And so we've been really interested in how this one receptor can facilitate both activation and immune evasion, and we sort of want to figure out how what exactly tips the balance with these pathogen interactions, and what it is particularly about an antigen structure and that relationship that is controlling uptake, trafficking, and signaling in dendritic cells. And so we wanted to develop probes to test this. And when you look at the glycans that are naturally present on a lot of these pathogens. So for HIV, one of the prevalent glycans is this highly branched MAN9. So you look at that, and that's pretty synthetically challenging, not super feasible to produce and test a lot of different things like we wanted to do. And so we wanted to find a more synthetically tractable alternative to probe these interactions. And for that, we turned to the use of glycopolymers, where we reasoned that with a polymer backbone, we could functionalize it to express a much more simple carbohydrate ligand, and then use the polymer backbone to mimic that multivalent display of carbohydrates that you see on pathogens. And so we utilize ring opening metathesis polymerization, or ROMP, to generate these polymers. And ROMP is really ideal for this because it's a living polymerization, and so you have much faster rates of initiation compared to propagation, and so you can get really homogeneous populations of polymers with very defined lengths. And then this chemistry is very gentle, and so you can incorporate a number of different epitopes and have really good control over the epitope density. To synthesize these polymers, I utilized an oxynorbornine monomer unit, and by varying the ratio of monomer to initiator catalyst, you can then control the length. And we incorporated an NHS ester reactive handle to install our desired ligands. And so from here, we needed to have a ligand that could target DC sign. And so we decided to use a mannose derivative, as mannose is a known ligand of DC sign. And so to synthesize this, I peracetylated mannose and then incorporated this phenoxyethyl bromide via glycosylation reaction, then substituted out this bromide for an azide, did the deprotection, and then did a final reduction to install this terminal amine, which we use to do our amide chemistry for functionalization. And so you'll notice that this ligand has this extra phenyl group incorporated that's not often found in nature. And we included this as a design feature of this ligand because, as with many lectins, DC sign has a lot of hydrophobic residues near the carbohydrate recognition domain. And so we reasoned that if we could incorporate this aromatic group, we could potentially engage some of those residues and increase the binding affinity to DC sign. And what we saw is with polymers that display just mannose versus this phenyl mannoside, we actually see about a 3- to 4-fold increase in binding with the phenyl mannoside, which is exactly what we were hoping for. So with this ligand in hand, we can then perform the amide chemistry to load it onto the polymers, and we can stoichiometrically control the amount of ligand that we want to put on. We also can incorporate a fluorophore for various cellular assays that we wanted to perform. And then we backfill all of our polymer functionalizations with ethanolamine to make sure that we don't have any remaining ester handles that could potentially do aberrant chemistry when we move to in vitro experiments. So to functionalize these polymers, one of our primary methods to characterize these polymers, our primary method is often NMR. And so we're lucky in that these backbone alkene protons are in a fairly distinct region of the NMR, and so we can integrate those. And then for this phenyl mannoside ligand, we also have some distinct proton peaks where we have this anomeric proton, and then these two phenyl peaks as well. And so we can integrate those and compare the ratio to the backbone alkenes protons to quantify the extent of epitope loading. And so we tested a lot of different densities of the ligand and experimentally determined that 35% loading of the mannoside ligand was about optimal for most efficient uptake. And as you start to increase that, you can occlude binding sites as it becomes too dense. And so we settled on this 35% loading for our future studies. And so the first thing that we wanted to do with our different polymer lengths functionalized with this mannoside ligand was confirm that we were indeed facilitating DC sign-specific internalization. And so we performed this uptake experiment where we incubated 2 different cell lines with our fluorescent polymers and imaged them by confocal microscopy. And so one cell line are these Raji cells, which are B cells that do not naturally express mannose binding receptors on their surface. And then we also had this Raji DC sign cell line that was stably transfected to express DC sign. And what we found is that only the DC sign expressing cells facilitate polymer internalization, indicating that it is going through this receptor. When we also investigate the uptake by flow cytometry, we can look at uptake as a function of manocyte concentration, where we're delivering the same amount of ligand, but are varying the valency with which it's displayed on the polymer. And so what we saw here is that with increasing polymer length and increasing valency, we actually saw increased polymer uptake and that efficiency. And so this is really important because many therapeutics that are out there today are seeking to target dendritic cells. And so we have this data showing that targeting these receptors with high valency materials, you can really get efficient uptake. From here, since we had validated it was going in via DC sign, we also wanted to investigate where these polymers were going. And so a colocalization experiment was performed where polymer, shown in green, cells were incubated with polymer and transferrin, which is in red, which is a marker of early endosomes, so that classical degradative pathway for immune activation. And then looked for areas of overlap as shown in yellow. And so what we saw here was that only the short 10-mer co-localized with endosomes, whereas these longer polymers were instead going to non-endosomal compartments that were near the cell surface. And so this was where I came onto this project. And so we were obviously curious about where these polymers were going. And so I was tasked with making more polymer links to further do more experiments. And so I came in and made these polymer links, but found something very different when I performed this colocalization experiment, which is that all of my polymers that I made co-localize in the endosomes regardless of length. And so this left us with this, very puzzling quandary of how are we getting these differential trafficking? And so I went through a lot of different experimentation, looking at making sure I had the same length polymers, the same densities, all of that. But what was really illuminating was when I looked at the physical properties of these polymers. What I actually found was that there were some huge differences in the size of these polymers, where the polymers that I made were all soluble in less than 10 nanometers in size, which you can see by DLS and TEM. They sort of ball up in solution to be about 10 nanometers or lower. Whereas the previous batch that was showing this aberrant trafficking had actually aggregated over time and was forming these larger particulate. And only the 10-mer, which remained soluble, was colocalizing in endosomes. So it was actually the size then that where small soluble polymers co-localize nicely in endosomes, where you can quantify that with Pearson's coefficient, where values close to 1 indicate high colocalization, and the aggregates do not, where values close to 0 are low colocalization. So from here, we wanted to see whether these aggregates were remaining in these compartments or whether they eventually make their way throughout this lysosomal pathway. And so we performed staining at different time points after incubation and looked at different markers throughout this endocytic pathway. And so what we found is that with soluble polymers, they continue to nicely co-localize through early endosomes, late endosomes, and into the lysosomes. And so this further, again, showed that we have these materials with high valency when they're soluble that we can really promote efficient trafficking to endosomes for degradation and antigen presentation. So that's very useful for informing dendritic cell targeting vaccine design. However, the aggregates seem to stay in these compartments, and as far as we've tested in terms of time points, they do not leave these compartments. They stay there. And so you can imagine the next obvious question that we had is, where are they going? What are these compartments? And so as we began to consider this, we were really reminded of the literature that's been published on HIV, where rather than going to endosomes, they go to these invaginated compartments at the cell surface. And so there's not really that much known about these compartments or about how HIV manages to get there, but there have been some preliminary studies that have characterized a bit of information about these compartments. And one is that while these are under the plasma membrane and the viruses are clearly inside the cell, these compartments are somehow surface accessible still. And so these viruses can easily pop in and out of these compartments. Additionally, studies have found that these compartments are enriched in the tetraspanin protein CD81, which is involved in HIV viral fusion, and so it just gets packaged along and is highly enriched in these compartments. And so we thought that we could utilize this information to begin to probe if our polymer aggregates were going to the same place. And so we performed this pretty simple but very neat experiment with trypan blue, where we incubated cells with polymer and imaged them before and after staining or treatment with trypan blue. And so trypan blue is typically a live dead marker because it cannot get into live cells, but it also has the property of being able to quench green fluorescence. And so the idea was that if these polymers were firmly inside and not in a surface accessible compartment, they would maintain fluorescence, but if they are surface accessible, you would see quenching of that fluorescence. And so that is indeed what we saw, where the soluble polymers lost very little to no fluorescence signaling, but with the aggregates we saw substantial amounts of fluorescence quenching after treatment with trypan blue. And so this led us to believe that these might be going to the same compartments as HIV. And so I endeavored to do further microscopy experiments to further characterize these compartments. And so the next two slides summarizes many, many months of work trying to optimize the staining protocols and the imaging techniques. But excitingly, what I was able to image was when I did co-localization experiments with either the two polymer batches, or the two polymer types, or HIV-1 virus-like particles, and co-stained with CD81, I was able to see formation of these CD81 rich compartments and nice co-localization with both HIV as well as with the aggregate polymers. Whereas for the soluble polymers, we see no co-localization and no formation of those pockets. Furthermore, I went another step further and performed direct co-incubation studies where I co-incubated cells with polymer and HIV virus-like particles and tried to see direct overlap. And so what I saw was with the soluble polymers, while they are spatially close, they seem to be in distinct areas of the cell, whereas with the aggregates, I was actually able to observe direct overlap. So you can look at this in terms of pixel overlap and see soluble polymers are in different space, but the aggregates, as far as the resolution of this microscopy allows, are overlapping with the HIV. So in summary for this portion, we have used polymers and looked at physical properties to see that we can get very different trafficking through the receptor DC sign. And so we imagine this project sort of splitting off into two very equally interesting directions from here. The first is that with these aggregate polymers, as far as we're aware, this is the first synthetic material that is able to access these same compartments that HIV is in. And so we envision that we can use these to further probe the mechanisms of how HIV is accessing these compartments. And then additionally, one factor that makes HIV infections very difficult to completely eradicate with antiretrovirals is the presence of these viral reservoirs in dendritic cells. And so while you can clear the active replicating virus, you still maintain these reservoirs that can be released at a later point. And so we think it would be really interesting if you could potentially use these aggregate polymers and see if you could displace the virus from these compartments to facilitate in treating these infections. And so that's with one direction with the aggregate polymers. And then with the soluble polymers, as I mentioned before, we developed this ligand and shown that soluble multivalent display of this ligand leads to really efficient uptake and trafficking to early endosomes for antigen presentation. And so we're applying this mannocide ligand and the optimal physical parameters that we've developed for a dendritic cell targeting vaccine. And so very briefly, we have switched platforms from polymers and have collaborated with MG Finn's group at Georgia Tech to use these self-assembling virus-like particles that can encapsulate immunostimulatory toll-like receptor ligands inside the particle, and then we can functionalize it with a high valency of this phenylmannocide ligand. And what we found is that this ligand on the particles targets them to DC sign and facilitates internalization into endosomes, and then additionally stimulates signaling that when antigen is presented, it signals for cytokine secretion to bias T cell differentiation into Th1 type cells, which are really important for cellular immunity against viruses and tumors. And so Murshida and the group is pursuing some really exciting work where he's taking these particles and applying them to a tumor challenge study to see if we can generate this kind of immunity to fight the tumors. So with that, I will switch into the second portion of my talk, which is focused on using polymers to inform vaccine design. And so as many vaccines that are out there function through the secretion of neutralizing antibodies, the focus is going to switch from dendritic cells over to B cells. And so, as I said before, we are a carbohydrate lab, and so sort of one of the impetuses that we had for this work is our interest in carbohydrate-based vaccines. And so carbohydrates represent a really promising target for vaccines because many pathogens express carbohydrates on their surface that are orthogonal to mammalian glycans. And additionally, tumor cells, because of their different metabolism compared to healthy cells, they actually overexpress specific carbohydrates. And so they're really a promising target for vaccine development. However, one thing that tends to hinder their use in this setting is that on their own, they are typically pretty poorly immunogenic. And so they have to be displayed on a really robust scaffold to generate protective responses against the carbohydrate. And so this is classically overcome through the use of protein conjugates, where multiple copies of carbohydrates are conjugated onto a protein carrier. And so here you get a multivalent display, which is important in B cell signaling. And then you also incorporate, in the protein, you have helper T cell epitopes that are also really important for robust T cell responses and help make this an immunogenic scaffold. And so while there have been protein conjugate carbohydrate-based vaccines developed, it is not a strategy that has worked for every pathogen or every carbohydrate. And so there's still gaps in the understanding in the field regarding these vaccine designs. And in particular, there's still not a complete understanding of the B and T cell epitope combinations and the display parameters that yield optimal responses. And so we hoped that we could contribute to the field here and see if we could do some systematic studies to gain a better understanding of some of these parameters to inform vaccine design. And so as we considered what kind of scaffold we wanted to use, there were certain features of protein conjugates that made them not really ideal for these sorts of systematic studies. The first being that these conjugation chemistries for the carbohydrate conjugations can be difficult and can often have low efficiencies. And additionally, there have been reports in the literature of off-target responses that are developed against the protein carrier or the linkers, and so you flood the system with those responses rather than against your carbohydrate. And so we really wanted to move away from using these sorts of scaffolds to do these studies. We thought that polymers offered a really nice blend of features instead to do these, where you can maintain a multivalent display and you can incorporate your carbohydrate, your helper T cell signaling, as well as additional stimulatory ligands in a very modular fashion where you can easily make a panel of different polymers to test. And as the backbones tend to be fairly inert, you can minimize some of these off-target responses. And so once again, we turn to the use of ROMP for its good control over length and the freedom with control over epitope density and different epitopes. And then as became important with this project in particular, you can also install unique end groups when you terminate the polymerization that can be really helpful in characterizing these polymers as they become increasingly complex. So as we began to think about what kind of parameters we wanted to look at, we thought about what was really important for robust B cell activation. And so there are two signaling elements that produce really powerful B cell activation. And the first is multivalent engagement of the B cell receptors by the antigen. And so that stimulates a primary signaling that starts a B cell towards activation. But to get really robust activation and antibody production, you need a secondary signal that comes from CD4 positive helper T cells. And so formation of that immunological synapse and that signaling really pushes the B cell towards activation, and you can get production of large quantities of antibodies that have gone through affinity maturation, so they're highly specific and have isotype switching, and you can also get formation of memory cells there. So for our initial studies, we wanted to use model antigens that looked at both B and T cell elements. And so we designed polymers to have a multivalent display of this model B cell epitope, dinitrophenyl. And we thought that with a multivalent display, you could cluster multiple B cell receptors, and that would trigger uptake into endosomes. Additionally, we incorporated a known T cell peptide epitope from the protein ovalbumin, and we linked it to the polymers via protease-sensitive linkage, with the idea that as these polymers are internalized and into endosomes, and as that acidifies, the protease cathepsin D would become active and would cleave this peptide from the backbone, free it for loading onto MHC and recruitment of that helper signaling. To synthesize these polymers, we utilized a norbornene NHS ester monomer unit, and again, by varying the ratio of monomer to initiator catalyst, we generated polymers of defined length. And then we could incorporate our amine-bearing ligands with stoichiometric control, and then again, we backfill with ethanolamine. Again, proton NMR was very useful in characterizing some of the epitopes, where we have the backbone alkenes, and then we have these two protons that are ortho to the nitro groups that are in distinct regions. And so we can easily characterize the extent of DNP loading on the polymers. However, the peptides become a bit more challenging because you have overlapping signals, and it can be really hard to isolate individual peaks with consistency. And so to get around that, we turned to the use of fluorine NMR, where we made this specialized fluorinated capping agent that we terminated every polymerization with, so each chain had that. And then for the peptide sequence at the N-terminus, we added a trifluoromethyl phenylalanine residue as well. And so then we can perform fluorine NMR and get really clean, beautiful spectra where our peptide peak is isolated and can be compared to the ratio of the backbone and cap. So with this synthetic scheme, we made a large panel of polymers that varied in the loadings of B and T cell epitopes, and we went into some in vivo mouse studies to look at B cell activation and antibody production, where we administered subcutaneous injections into mice at day 0 and at day 34, and then did weekly blood collections and analyzed the serum for DNP-specific antibodies using an ELISA-based assay. So one of the first things we wanted to look at, as I mentioned in my first talk, we've seen that multivalency is very important in a lot of immune cell responses. And so we wanted to look at the differences between a short and a long polymer with increasing valency of the B cell epitope. And so we immunized mice with a 30 mer and a 300 mer, and saw that with the higher valent of B cell epitope display, you see enhanced activation of B cells and isotype switched antibody production. And so this experiment was performed where we dosed each mice with the same number of polymer chains between the two different polymer lengths. And so one thing that we had considered was that for the longer polymer, you're potentially just administering more DNP since you're going at a per chain basis. And so we wanted to ensure that this result was in fact due to valency and not due to just the increased DNP concentration. And so we performed an experiment where we did the same dose of 300 mer and 30 mer at the same number of polymer chains, but then dosed with more 30 mer to equal the same DNP concentration as the 300 mer. And what we saw is that still with the 300 mer, you see the most robust responses, indicating that this is an effect of valency. In addition to looking at B cell epitope valency, we wanted to explore T cell epitope valency. And so we immunized mice with polymers that had the same length and the same amount of DNP loading, but varied in the amount of the T cell OVA epitope loading. And what we observed is that with the polymer that had no T cell epitopes, it was T cell independent, you do still see formation of IgG titers specific to DNP, and so that is indicating that the multivalency is sufficient to induce some. But as you increase your T cell epitope loading, you see marked increases in the antibody titers, indicating that a multivalent T cell epitope display is also very beneficial for these responses. And this is very important for the field, as many of these vaccine formulations don't tend to include quite this number of T cell epitopes. And so we think that this could be really helpful in future vaccine designs to boost the number of T cell epitopes that you're incorporating. In addition to looking at the number of T cell epitopes, we wanted to investigate the inclusion of this controlled release mechanism that we had engineered into the T cell epitope and see if we were seeing any enhancements there. Again, because many of these T cell epitopes are either included in the protein or they're just linked directly onto the scaffold. And so we wanted to see if this could afford enhancements as well. So we immunized mice with 300 mer polymers that had the same amount of DNP and OVA, and the only difference was that one polymer had the cathepsin D sensitive linkage and the other had the cathepsin D insensitive linkage. And what we saw is that you do see some enhancements with that controlled protease release mechanism, indicating that this, again, is a unique design principle that can be broadly applied potentially to vaccines. So with these polymer panels that we tested, we wanted to see with our optimized polymer scaffold how that compared to a comparable protein conjugate and see if we were indeed seeing enhancements in responses relative to that. And so we immunized mice with two different polymers, both 300 mers. One only had B cell epitope and one had multivalent B and T cell epitopes, and then compared it to DNP ovalbumin conjugate where the ratio of DNP to OVA was the same as it was in the polymer. And indeed, we saw that with our optimized polymer scaffold, we did see enhancements in the responses, indicating that these could be some generalizable principles that can really assist in vaccine design. So with this exciting data, I wanted to further investigate the fate of these polymers in vivo and look at things like how they're being cleared and their toxicities. And so I performed some experiments in mice where I immunized them with a short 30 mer or 300 mer, and then did some histology of these different organs at different time points, and was excited to see that compared to a healthy mouse with both polymer treatments, we didn't see any excess inflammation, no tissue distortions, and we detected no toxicity, which was really exciting. Additionally, I wanted to look at the distribution of these polymers in vivo, see the rate at which they were clearing, where they were going. And so again, I tested mice with a short and a long polymer and saw that for both polymer lengths, we see a majority of the clearance through the liver. And so this is not super surprising to us because if you remember from the DLS data from the first portion of my talk, the size of these polymers doesn't necessarily increase linearly with length. They do tend to ball up, and so a 30 mer and a 300 mer are actually not that much different in size and solution, and so it makes sense that they're clearing through the same mechanisms. Finally, I wanted to investigate some of the pharmacokinetics to see how fast it was clearing from the site of infection. And so I tried to perform these studies by doing fluorescence analysis of the blood, but was unable to detect any fluorescence at any of the time points we looked at. And so this suggested to us that potentially from the subcutaneous injection site, we were seeing a slower release of these polymers, and it wasn't able to build up to detectable amounts. And so I performed this pseudo-pharmacokinetic experiment where I injected mice with fluorescent versions of these polymers and then performed live imaging of the next graph to image and quantify the rate of diffusion of that fluorescence signal. And so while this isn't the whole picture because you're only getting the surface and you don't have any information about the depth of that fluorescence, but generally what we saw is that for both polymer lengths, In about 6 hours, you see loss of about 50% of your fluorescence signal, but after that, you start to see a decrease in the rate of diffusion, indicating that these polymers are potentially being released slower, so whether they're forming a depot at the site of injection and slowly releasing. But this is really interesting to us and has sparked a lot of thoughts about future experiments looking at different routes of injection, as that can have a big impact on vaccine efficacy. As well as with this slow release, there have been studies in the literature that have shown that smaller dosage that is over a longer time can actually more mimic the natural exposure to a pathogen and actually improve responses that way. And so there's a lot of really interesting directions I think we can go with this, looking at dosing and route of injection and pharmacokinetics. But broadly, in summary, with this project, we've used polymers and have showed that a multivalent display of both B cell epitope and T cell epitope, as well as the inclusion of a protease-sensitive release mechanism, can lead to increased B cell activation, and that when you compare it to a protein conjugate, we do see enhancements there. And so from here, we've been really excited to take these studies and test them with some more physiologically relevant antigens. And so one project that Laura alluded to in her intro is this work with the chickens that for time's sake, I don't have time to go into it fully, but I'm absolutely happy to talk about it after my talk. But we've been interested in taking what we've learned with our polymer scaffolds and applying it to a vaccine design that has been seeking to eliminate the use of antibiotics in agriculture and replace them with antibodies. And so there's a big push in the field to get rid of the use of antibiotics because it's led to the generation of antibiotic-resistant strains. And so there's a push to get rid of them, but you also need to still find ways to therapeutically treat infections in poultry and cattle, and also potentially promote weight gain. And so we had the pleasure to work with the Cook Group at the University of Wisconsin to try to improve on their method where they vaccinate laying hens with a protein conjugate that is specific against an intestinal target, because a lot of infections that are common in poultry and cattle are based in the intestines. And so they can vaccinate these laying hens against those targets, and then the hens lay eggs, and in the eggs are large quantities of antibody that is specific for the epitope that you're vaccinating against. And so you can dry these eggs down, spray it over the feed of the animals, and directly deliver those antibodies to the site of infection. And so the problem with this currently is that their method is too expensive to be broadly applied commercially. And so we've been working with them to, where I've made a lot of different polymers, and we've tested these constructs to try to improve the antibody yields that you're getting to drive that price down. And so I have some really exciting data where we've found a polymer scaffold that can prolong the antibody titers, and we hope that this can be really transformational in getting this commercially applied. So in addition to the animal health vaccine applications, again, we're really interested in these carbohydrate-based vaccines, and so there'll be a lot of future work, I think, in the group where we want to see how generalizable these principles are of design and apply them to some really relevant carbohydrates in pathogens that are still in need of effective vaccines. So in conclusion, I hope I've showed you the utility of using polymers to investigate different facets of immunity. And we have found that a high valency display of a dendritic cell targeting ligand can enhance efficiency of uptake and endosomal processing. We've also found that antigen size can have huge impact on uptake and trafficking through the receptor DC sign. And we now have synthetic materials that we hope to be able to probe these HIV immune evasion mechanisms. And then alternatively, we've also found that a multivalent B and T cell epitope display can enhance B cell activation and antibody class switching and production. And so we're really excited about the directions that we have to apply these polymers to vaccines for animal and human health. And so with that, I have a whole load of acknowledgments. Laura, I have to thank you first. I'm just so grateful that you took me into your group and for all the support you've provided over the years, really pushing me to think independently about these projects and sort of take ownership and take them in directions that I'm interested in, even when it meant vaccinating chickens, which was a little weird. But I just feel like I've grown so much under your mentorship as both a scientist and independently. I'm just incredibly grateful to you, so thank you. I also want to thank my thesis committee, Professor Shalik and Wittrup, for agreeing to serve on my committee and provide feedback on my thesis research. Within the Kiesling group, I have to thank Joe Grimm and Danny Zwick, who I worked with closely on the polymer aggregate project, and Natasha, who I worked with on the B-cell mouse work and really laid a lot of the foundational groundwork for these in vivo experiments and performed a lot of them as well. And then Rashid and Austin and Helen have been really excellent collaborators in the group, and I've been really fortunate to work with them, and I've really enjoyed all of our scientific discussions, and I appreciate all of your contributions to pushing these projects forward. And then I had a really great undergraduate at University of Wisconsin, Angie Jasper, that I was really fortunate to work with as well. In terms of collaborators, the Shearer group, and particularly Jay and Lorraine, they provided the virus-like particles, the HIV virus-like particles for the first part of my talk. The Johnson group and Hung really helped me get the biodistribution and toxicity studies going in the mice. And then Mark, I'm really grateful that I had the chance to work with him, and he was just a really great collaborator, and he's greatly missed, but I think we've been really fortunate to have the opportunity to have worked with him while he was here. And I have to thank Maria Arendt, who was really the force behind getting the chicken work done even after Mark's passing. So I'm incredibly grateful for her to getting that project to a point of completion where we could finish out that work that Mark was excited about. The core facilities at both MIT and at Wisconsin have been really tremendous in helping me with a lot of different experimental optimizations. Wendy Salmon and Nikki Watson at the Whitehead here were really helpful in optimizing the confocal experiments and the TEM. And then in the DCIF, Walt was really instrumental in getting the right program parameters for these polymer characterizations. The same for Charlie at UW, and then Martha for help with mass spec. So something that I've been asked before is, are you part of an awesome lab group? And the answer to that is absolutely yes. I have been incredibly fortunate to be a part of the Kiesling group, who have stimulated me both intellectually, but have also just been a great support system. You guys have contributed a lot to being there for just off-the-cuff scientific discussions, as well as really helpful contributions during group meetings and the like, but you've also just really made it a great environment to work and to come to work every day, where I know that I will be challenged scientifically, but also be incredibly supported and just have a lot of fun doing science that we all enjoy. So thank you all for that. I also want to thank a handful of friends. We have been really fortunate in that we've had experiences both at Wisconsin and MIT in that academic setting, and I've also had the opportunity to make some really great friends that have stuck with me through kept in touch throughout the move, and then of course our trivia team in Wisconsin was really great. But then with the move, I've also had the opportunity to have some really great new experiences with old friends from Wisconsin, as well as meeting really exciting new people here as well. And so I learned how to ski out here, which was very exciting and fun to do with friends. And then I also was fortunate to have a really excellent mentor group through the graduate women at MIT that I'm very thankful for their support over the time that I've been here. Lastly, I have to give a huge thank you to my family, who I owe so much to, and have really made a point to come and visit me even after moving here, and some of them made the trip out here just especially for my defense. And so I have to thank my dad and my stepmom Becky, who were not able to be here today but have still been a constant source of support from afar. I want to thank my friends Katie and Zach, who are here today. And they were so gracious to come and celebrate with me. Katie and I met in undergrad, and while we technically aren't family and blood related, I definitely consider you as part of our family, and I feel like we integrated well. So thank you for all of your support over the years, and for coming to celebrate with me today. To my mom, who is also here, you have always encouraged me to follow what I'm passionate about and have been there to really invest in my work, and whether it's asking me like, what did you do today? Were you working with the chickens or the mice or the microscopy? So while we might not have gotten like the scientific depth, you're still very invested in that, and I really appreciate that. And thank you for being there to encourage me through the hard times when I was, hitting a wall in terms of research, but also celebrate when I've gotten things to work. And then lastly, to my sister, who from the very beginning, we were best buddies and partners, and not much has changed over the years. We're still a dynamic duo that just dresses up like Power Rangers a lot less. But I really appreciate all of your support and your sense of humor. I feel like it matches mine, and you always know when I need encouragement or support or just celebration. So thank you to all of you for being here. And with that, I would like to thank you all for your attention, and I'm happy to answer questions.
Okay, any questions from the audience?
I have a question that has been messaged in from one of our live stream viewers. This question is from a research scientist at Kaleido, who asks, does the slow clearance from injection site in the mice studies suggest that low responses in chickens using polymer for initial challenges might be related to low systemic doses? Does that make sense? I think I said that right.
So. So it's certainly a possibility. We haven't been able to look at any of the clearance or in the chickens. We do inject intramuscularly, which also can have a difference, but it's possible. I think what's the more likely scenario is that the protein conjugate that they use it can aggregate and serve as like an additional immunostimulant, and so I think that it's just much better at priming the birds because it has that extra adjuvancy with the aggregation. But I think the short answer is yes. I think it could play a part and could potentially be optimized for better drainage and optimal B cell activation.
Thank you.
Yeah, Roger.
So I noticed that back with Natasha's work, one of the issues was that the polymers gave, when you re-challenged the mice, they gave a stronger response to the T cell than the B cell epitope. Do you still see a lot of immune memory and immune response to the ova itself?
In terms of B cell activation, or like, what do you? I guess
in
I don't
terms
understand.
mostly of antibody production.
Oh, against ova. So we didn't look into antibodies against ova in these studies. That's not. No, sorry. We did a small study where we isolated splenocytes, and then we looked at antibody-secreting cells and coated a plate with the ova peptide, and we don't see any. We see little to no production of ova-specific antibody-secreting cells, and so I think that's not a huge issue that we've encountered in the mice studies. That answers your question, I think. Yeah.
For the DC sign that was activated uptake, so you're using the phenylamidoside ligand to like get that DC sign signaling. Is there any concerns since that did increase the binding affinity? That if you're like looking for a natural nanoligand, that whatever like response you're seeing with the polymer like like phenylalanidoside, it's not going to be like mimicked anymore with natural nanoligand?
So we have evidence that suggests that the aggregates with this ligand on there signals in the same way that like HIV does, and so we seem to be still mimicking that same signaling. I can't say whether that's true for like every mannose expressing pathogen, but the ones that we've looked at, it seems like it's still it's just a way, because we don't have quite the like branching and quite the extension of those ligands as the natural pathogens do. We can hopefully kind of like get up on that part of it with increased affinity of the ligand and sort of match. And it seems like we sort of have achieved that, at least in what we've studied. So yeah.
How can you backfill with ethanol and not just like ammonia?
And not just what?
Not just like ammonia or something.
I think it was just, I don't know if there's like maybe Laura can choose the primary one, but
I think actually ammonia isn't that nucleophilic, and so anything else you'd use, you have to use like a different amine, and you might add more hydrophobicity. So we were trying to avoid adding anything hydrophobic.
and it's like pretty cheap, and you can just add a lot of excess of it, and it doesn't seem to impact the overall structure of the polymer. So it's I'm not sure about like initial rationale, but as far as like what we've been using them for, it seems to be like pretty inert and a quick way to just make sure you don't have any esters.
We tried something called glucamine, which is just if you do a reductive amination of glucose. So you have a polyol to change the solubility of the polymers as well. So we played with that a little bit to change solubility. But ethanolamine works better. You'd think that glucamine would be more polar, but it actually caused the polymers to aggregate.
Yeah.
You talked about the aggregated polymers maybe having the potential to like flush HIV out of those compartments. Have you thought about any other materials that you might use other than aggregated polymers for that job?
Yeah, so these are, like, we access them, like, you know, as I mentioned, sort of serendipitously, and so we decided to, like, you know, I developed, you know, ways to take soluble polymers and intentionally aggregate them, and it was just an easy way to continue looking at the interesting biology. But I think moving forward, this project would absolutely benefit from a much more defined polymer scaffold, or like a liposome of sorts, where you can really control the size and the display of the mannose, because we don't really have a great understanding of how that mannose is displayed. And so I think being able to use materials where you can control size and density of display and potentially even rigidity can sort of start to tease apart the individual features that contribute to the trafficking phenotype.
If there's no further questions, let's thank Cassie again, and we'll go to close session.